Zullen we gaan staan? U kunt op plaats.
Can I start? Okay. So, dear Prorector, members
Sorry, Francesco, this is my line. I have to officially open this ceremony, but first of all, I would like to welcome you all. Family from Italy, I understand. Welcome. For this ceremony, in which Francesco Schianchi will try to defend his academic thesis entitled "From v-ATPase to Protein Palmitoylation, Novel Targets Against Lipid-Induced Cardiomyopathy." Before we go to the opposition, Dr. Schianchi, Mr. Schianchi, I'm already anticipating. I would like to ask you to present a brief overview of the results of your work and the major conclusions in about 15 minutes time. Please.
Okay. So, dear prorectors, member of the Corona, family, friends, and colleagues, in the next 15 minutes, I will shortly give you an overview of the research I've been done during my PhD, which is described in the thesis entitled "From v-ATPase to Protein Palmitoylation, Novel Targets Against Lipid-Induced Cardiomyopathy." So the organ object of this study is the heart. Before we go any further in this presentation, it is important to understand how the healthy heart is provided with enough energy to function properly. So you have to think that the heart is like a car. They both need fuel to function properly. So a car needs fuel to go on the road, to speed up on the road, and the heart needs fuel to pump properly inside your chest. The fuel that the heart uses to do that are fatty acid and glucose. More importantly, the heart has to use them in a balanced manner. And a healthy diet is of high importance for providing the heart with a well-balanced amount of energy substrate to maintain proper cardiac functioning. However, overconsumption of fatty acid, as it happens during with Western diet, will cause a disbalance in cardiac energy substrate.
Schianchi, can you stand back a little bit? Yeah, so otherwise the...
Okay. So will cause a disbalance in cardiac energy substrate, and this will cause cardiomyopathy and decrease of heart function. So cardiomyopathy is defined as abnormal cardiac structure and performance in the absence of other risk factor, and is a major complication in diabetes. So diabetes is a big problem of today, and in 2021, 537 million of people suffer from diabetes, and this figure is set to increase to 783 million in 2045. Moreover, 16% of diabetic patients have 16% higher risk of developing cardiomyopathy compared to healthy subjects. Cardiac insulin resistance is the main factor for cardiomyopathy. So normally insulin stimulates the translocation into the heart of the fatty acid. Also, insulin stimulates the uptake into the heart of glucose. And this happens through the glucose transporter for glucose and for the fatty acid transporter that you see in green in the screen. Cardiac insulin resistance occurs when excessive amount of fatty acid enters the heart, and this will inhibit the action of insulin on the transport of glucose. But how this process is regulated inside the cell at molecular level? Obviously, the heart is not obviously the heart is not happy when like the glucose doesn't enter into the cell, and there will be more fatty acid that comes in. So at molecular level, this process is regulated as follows. So here you have your heart cell. This is a schematic representation. And in pink, you see the cytosol. So the pink region is the cytosol. The gray region is the cell membrane. Inside the cell, you will have some vesicles that contains the glucose transporter. There will be other vesicle that contains the fatty acid transporter. And on the extracellular environment, there will be the blood vessel where insulin hormone flows. Hopefully, you will have also some glucose and fatty acid that you ingest with the diet. And on the surface of the cell membrane, there are some receptors for insulin. So after a meal, what happens is that when insulin binds to this receptor, it will stimulate the translocation of the glucose transporter from the vesicle to the plasma membrane, and this will cause the uptake of glucose inside the heart. Insulin also partially regulates the translocation of the fatty acid transporter from the vesicle to the plasma membrane. But this process is further regulated by another protein called v-ATPase that is formed by these two parts, so that you see in green on the screen. When these two parts are assembled together, they can further regulate the correct trafficking of the fatty acid transporter to the plasma membrane together with insulin. And this causes the fatty acid to enter into the heart. So there is a balance in cardiac energy substrate, and the heart can beat properly. But fatty acid oversupply causes this protein, v-ATPase, to disassemble. So this protein cannot regulate anymore the trafficking of the fatty acid transporter to the plasma membrane. This process is not regulated anymore, and this protein will start to accumulate at the surface. So more and more fatty acid can enter the cell. They will accumulate inside your heart, and this will inhibit the transport of glucose inside your heart cell. So glucose cannot enter your heart anymore. There will be a disbalance in cardiac energy substrate, and cardiomyopathy are likely to develop. In this condition, your heart is insulin resistant. In this scenario, we don't know, we miss some pieces. For example, we don't know how fatty acid can assemble v-ATPase. We don't know how fatty acid inhibit the uptake of glucose into the heart. In this thesis, we also aim to reassemble v-ATPase through some pharmaceutical approach. In this way, the translocation of the fatty acid transporter can decrease, and fatty acid can start to accumulate less into the cell. So in chapter 2, we try to answer to the first question, so how fatty acid induce v-ATPase disassembly. In chapter 4, we investigate how fatty acid inhibits the uptake of glucose and cause insulin resistance in the heart. And in chapter 5, we aim to reassemble v-ATPase. For chapter 2 and 4, we have a common hypothesis. So we hypothesize that fatty acid induce v-ATPase to disassembly and inhibit the uptake of glucose through a process called protein palmitoylation that I will briefly explain in this slide. So we know that the most abundant fatty acid that you absorb with your diet is palmitate. So palmitate, you can see, is this orange stripe is represented by this in this way in this slide. And when it enters the cell, it can directly bind to protein. So palmitate is able to stabilize proteins within membranes and can regulate some process, such as recycling of protein between intracellular compartment, between cytosol and plasma membrane, in this process that is called indeed palmitoylation. Is important for cellular processes such as uptake of nutrients. Palmitoylation also influences protein function. It can assemble two proteins, thus ensuring correct protein function. fatty acid oversupply causes intracellular increase of palmitate, which will bind more and more to protein. So the protein will be more palmitoylated, and this disrupts normal function of protein, such as the uptake of nutrients inside the cell. More palmitoylation also causes protein to disassemble. So it's important and will inhibit protein function. So it's an important process for proper function of protein. So we hypothesize that increased intracellular palmitate concentration will bind to protein, will cause more palmitoylation of protein, thus inhibiting glucose uptake or augmenting fatty acid transporter translocation to the plasma membrane and augmenting fatty acid uptake into the heart. This will cause cardiomyopathy and insulin resistance. So the first candidate that we investigated was v-ATPase. So we thought that increased palmitoylation of v-ATPase would disassemble this protein, thus causing the increase in fatty acid uptake that we observe. In chapter 2, we explored this hypothesis. So we investigated if this protein was palmitoylated, but we didn't find any palmitoylation of v-ATPase, unfortunately. So we could conclude that fatty acid do not induce v-ATPase disassemble via palmitoylation. We were a bit disappointed, let's say, but we kept going anyway. And we investigated some other protein, which palmitoylation we thought was involved in the inhibition of glucose uptake. So in chapter 4, we investigated that. We used some cell from some heart cell from the rat. We use a special equipment that analyzes all the palmitoylated protein into the heart. So we got this big list of protein. We selected 3 of these protein out of this list that we know that are involved in the regulation of glucose uptake. One of them was IRAP. So this protein is palmitoylated, and inside the cell, the function of IRAP is the following. This protein interacts with the glucose transporter, and when insulin binds to the insulin receptor, will cause translocation of these 2 proteins at the plasma membrane so that glucose can enter into your cell. We found that oversupply of fatty acid increased the palmitoylation of IRAP, thus inhibiting the function of this protein and inhibiting the translocation of the glucose transporter to the plasma membrane. So glucose cannot enter your heart cell anymore, and your heart will suffer, unfortunately. There will be insulin resistance and cardiomyopathy. We found also that inhibition of this excessive palmitoylation of IRAP rescue the translocation of the glucose transporter and the glucose uptake inside the cell. Cardiac energy balance is restored, and also heart functioning are restored. So our conclusion is that increased palmitoylation of IRAP impairs the uptake of glucose in the heart. This will cause insulin resistance and cardiomyopathy, and that targeting IRAP palmitoylation could restore glucose uptake and cardiac function in the insulin-resistant heart. So our heart is happy again. In the last chapter, we try to reassemble v-ATPase through some therapeutic strategy. So we thought that amino acid supplementation was a suitable option to do that. So why amino acid? Because we know from some previous research that amino acid can interact with v-ATPase, thus hopefully reassembling this protein and rebalancing fatty acid uptake and restore cardiac function. So in this chapter, we saw that supplementing the cell with a specific amino acid cocktail could reassemble v-ATPase, thus restoring correct plasma membrane translocation of the fatty acid transporter. Less and less fatty acid will accumulate into the heart cell, and this will relieve the inhibition on the transport of glucose inside your cell. So glucose can enter again your cell, and there will be restoration of cardiac energy balance and also restoration of contractile function. So our conclusion for this chapter is that amino acid supplementation can restore v-ATPase reassemble and also contractile function. So amino acid is an effective strategy to restore cardiac function in the insulin-resistant heart. And your heart, again, is happy again. As a summary, we can say that v-ATPase and IRAP are suitable targets to restore contractile function into the heart, and that v-ATPase and palmitoylation are novel targets against cardiomyopathy induced by overconsumption of fatty acid. And I want to thank you for your attention.
Thank you, Mr. Schianchi, for this very clear overview and very nice animations showing and explaining these very complicated processes during cardiac energy uptake. Your supervisors have chosen a team of experts first to assess the quality of your thesis, which was positive. And, but there are questions that remain, of course. So they have added some extra members to this opposition committee. And we will start the opposition now. And Professor Brouwers will start the opposition. Professor Brouwers is professor of internal medicine, more particular endocrinology and metabolic diseases at this university. Professor Brouwer.
So thank you. Well, dear candidate, before I start off with my questions, first I would like to congratulate you with this very nice piece of work. I enjoyed reading it. Sometimes it was a little bit complex for me as a clinician, but still I enjoyed it. Also would like to congratulate your supervisory team with this very nice result. I would like to elaborate with you on chapter 5. I think you just nicely showed what you did in your experiment. You gave a cocktail of amino acids to these cardiac cells, and you showed that the function of these cardiac cells, or in these rodent models, that it improved, and that this improvement was mediated by an mTOR activation of this v-ATPase assembly. I was a little bit puzzled by that because in the diabetes field, and you mentioned a little bit on that in your discussion, but in the diabetes field, it has been suggested that activation of mTOR is not that beneficial. So could you comment on that?
Highly esteemed opponent, thanks for your question and for your kind words. I understand your question, but so what we observe is that in the heart, mTOR, it stays always on the membrane of the lysosome or endosome, where it is in complex with v-ATPase, lambtor, or this big complex of protein. So in the field of diabetes, it's suggested that indeed can inhibit insulin signaling because it translocates to inhibit the insulin receptor substrate. That's what we don't see in the heart, so we see that it always stays on the membrane of the lysosome. Yeah,
so that's what you say, and you refer to figure 2 in that chapter to confirm that. Perhaps I missed it, but did you perform these experiments of where this mTOR was located also with co-incubation of insulin? Is there an interaction of these amino acids with insulin on the localization of mTOR?
With insulin, now that I recall, no, we didn't perform.
So you don't know exactly if you give the amino acids, mTOR stays, but if you give insulin, it gets removed from the membrane. But what happens if you give them together? You don't exactly know.
Together with insulin, I think it's a good suggestion. Mm-hmm. So I think we should also explore that. But no, we didn't explore this. So we didn't explore the localization of mTOR together with the stimulation of insulin.
Yes, because I read the chapter, of course. You did additional experiments. You continue your chapter with performing experiments with insulin. So if I look at your figure, what is it? At least figure 4, also figure 3. But when I look at figure 4, you did some co-incubation with insulin. And so I was just wondering, what do you conclude from that? If you look at your results, and if you give this cocktail of amino acids and you co-incubate with insulin, what is your conclusion based on that?
Thanks for your question. And so for this, for this result, our conclusion is that when you give insulin, for example, in high palmitate, when you give insulin, the uptake of glucose, for example, is inhibited, but when you add rapamycin, which is the inhibitor of mTOR, you have a little bit of decrease in the uptake of glucose, meaning that in some way v-ATPase is involved in this, process.
So what I think concluded from figure 4, I guess F, if you give this high fat diet, that with insulin you see an increase in glucose uptake, which is abolished without the amino acids. So apparently, if you give the amino acids with insulin, it's good. It's good for the glucose uptake. So apparently there is no true interaction.
Yeah, apparently it's good.
Okay. And then I hope to read a little bit more on this in your general discussion, but you decided not to discuss on that. But luckily, in your impact paragraph, you noted that a clinical trial is in preparation with regard to amino acid supplementation and what it does on cardiac function. Is that study already ongoing? I'm very curious about the design of the study.
Yeah, so we have our former colleague that now is in China is following up this work and is also set to give this cocktail of amino acid to patient.
Yeah. Do you know if there are already studies that were performed on amino acids and cardiac function, but perhaps in even a little bit broader perspective, insulin resistance or diabetes?
From what I know, they want to focus only on the heart, so to improve specifically cardiac function. So
I noted in your slides a happy heart, but it should also be a happy patient, of course. So we try to treat the whole patient.
Hopefully,
so,
yes.
But are you aware of any studies that because I think somewhere in your thesis you also mentioned the fact that it's also, by the way, one of your propositions that if you improve the heart, probably you also improve the muscle. Yes. So, and muscle is a major organ where insulin resistance whole body takes place. So, are you aware of any studies where they supplemented amino acids and what it did on insulin resistance?
Yeah, I read something about that, and indeed, there are some studies that they say that it improves insulin resistance.
Okay, because at least I think from our own university, there's been Dr. Felix from another department who tried to lower amino acids, specifically the branched chain amino acids, and what she noticed when she administered a specific drug to lower the branched chain amino acids, that insulin resistant improved. So the other way around. So I'm so much curious about the study that will be conducted, what the results will be of that.
Yeah. Can you briefly answer the question? Briefly?
Yeah, I'm also curious because indeed it offers the possibility also to target organs. So because this specific cocktail of amino acid target the heart, but maybe the same mechanism that we see with v-ATPase is also in skeletal muscle.
Yes, or the opposite. I'm very happy with the answers that you were given. I will give the word back to the prorector.
Thank you, Professor Brouwers. The opposition will be continued by Dr. Heather. Dr. Heather is a specialist in cardiac metabolism at the Department of Physiology, Anatomy and Genetics at the University of Oxford. The floor is yours.
Thank you very much. Very much enjoyed reading your thesis, Francesco, and thank you for allowing me to be part of this committee here today. I wanted to kind of delve into chapter 2, if that's okay, and the v-ATPase and palmitoylation. And I wondered if you could kind of talk us through kind of your hypothesis and your theory as to why you thought the v-ATPase was going to be palmitoylated in the first place.
Highly esteemed opponent, thanks for your question. So we choose v-ATPase because we know already that, for example, in neurons, v-ATPase is palmitoylated, and in some diseases that I don't recall the name now, but persistent v-ATPase palmitoylation cause misplacing of this protein, and the lysosome cannot acidify properly anymore, and this causes some kind of disorder that I don't recall, but I know that it causes this not acidification of the lysosome. So that's why we thought that also in the heart could have happened the same, and also because in the brain there is the same subunit. So there is a subunit, that subunit A, I think, of the v-ATPase, and there is also in the heart, and we thought that maybe could have possible.
Okay, so carrying on from that, how do we then rationalize the fact that we've got something very different going on in the neurons compared to what you're seeing in the heart? What explanation can we therefore form?
But this is very interesting question, because I'm also interested in that, because we observe different things, and this can mean that palmitoylation has different function in different tissue. So somehow this modification helps v-ATPase in the neuron, but in the heart not. So there is a specific regulation. This is very interesting.
Okay, so if we kind of take a step back and we think about the process of palmitoylation, in a lot of chapter 2, you're talking about lipid overload and potentially driving more palmitoylation. But what other factors determine whether a protein is palmitoylated, apart from the supply of palmitate?
So there are some enzymes that mediate this process. So, and there are 21 isoforms of this enzyme, and this can also explain why, for example, in one tissue, one protein is palmitoylated and in another tissue not, because these isoforms are expressed in a very different way between tissue. Depends on what is the use of palmitoylation in that specific tissue. So.
And what about depalmitylation? Because it's depalmitylation. You know, we've got to look at both sides of the balance because we have flux in both directions. What kind of fact? Depalmitylation.
Okay, sorry.
Right, in my accent. Depalmitylation.
Depalmitylation. There are way less enzyme compared to the palmitoylation enzyme. So there are only 3, 4 maybe enzymes that are known, and seems that they have less specificity compared to the palmitoyl acetyltransferases to the DHC.
Okay, so you think the specificity is in the process of adding the PTM rather than the process of removing the PTM?
Can you repeat your question?
You think the specificity lies in adding the palmitoyl-CoA on rather than the specificity of removing the palmitoyl-CoA?
Yes.
Okay, so in your lovely presentation at the beginning, you were showing us that palmitoylation is specifically involved in kind of moving things to membranes.
Yes.
So going back to your initial theory, you thought that palmitoylation would be involved in removing the v-ATPase away from the membrane.
Yes.
That seems counterintuitive to me.
Yeah, I can understand. So was mainly done this animation for like animation purpose, but it's more complicated than that. Palmitate can anchor proteins within membranes, but it can also kind of disassemble protein independently if they are on the membrane or not. So especially if it's a big complex, that can happen also, as it happens for v-ATPase in neurons.
Okay, so my final question relates to the bigger, I suppose, field of palmitoylation and these different experimental approaches we can take to look at palmitoylation. And you use the ABE approach in your experiments, the biotin approach. And one of the big issues is, how do we rationalize results from those approaches, from things like the Swiss Palm data sets, which are showing huge numbers of proteins palmitoylated, and experimentally they're not being validated? How do we see the field moving, and how do we rationalize why different studies are showing different targets that then can't be validated?
Okay, yeah, I understand your question, but you mean different target in the same tissue, for example?
Quite often, yeah. So like things like the data sets are showing far more proteins than we're ever being able to validate. How do we explain that?
It can, in my opinion, depends also on the assay, for sure, because it's a quite accurate assay, but depends on how you perform, the condition, the initial amount of protein that you use, it can give different results. So I think it's useful, in my view, like for example, for one tissue, it should be standardized. I don't know, the amount of protein that you use, I know it's difficult, but in my opinion is this that makes the difference.
Okay, thank you. That is all my questions.
Thank you.
Thank you, Dr. Heather. The opposition will be continued by Professor Sluiter. Professor Sluiter is present online, and his expertise and chair are in cellular and translational cardiology at the University Medical Center of Utrecht. Professor Sluiter, the floor is yours.
Thank you, Mr. Chairman. Dear candidate, I hope you can hear me. Just waiting for you.
Yes. Can you hear me?
Just to be, yeah, it's perfect like this.
Okay.
Sorry for not being there. And there's some echo, so I'll try to cope with it. But I would like to start, of course, to congratulate you with this beautiful work provided in this extensive thesis. I really enjoyed reading it, although it's outside of my expertise field in this molecular level. And I need your help coming, 6-7 minutes to explain me a little bit more on the details there. In my words, I also like to include your supervisory team, family and friends, of course, to congratulate them, because probably you have to explain definitely your friends a lot on what you did in the lab. So, so the impact that you describe on the diabetic field, the insulin resistance, the problem that we're facing. Myself also, I'm working on the genetic cardiomyopathies. So what I hear from you explaining in detail on the mechanisms and the underlying problems, I hear several things as well overlapping with the genetic cardiomyopathies. So you have this problem with the fatty replacement that comes into the cell more than the glucose. It's similar in heart failure and also genetic cause. The lipofuscin that's like aggregates forming in these cells. So for me, it's overlapping. So how much is the diabetic cardiomyopathy so different than the other cardiomyopathies like the genetic background? Maybe a big question, but looking forward to your answer.
Highly esteemed opponent, thanks for your question and for your words for the thesis. So the diabetic cardiomyopathy are indeed more influenced by increased uptake of fatty acid compared to other to the genetic cardiomyopathy, if I understood your question, or?
Yeah, that's correct, but I'm not sure if it's so different, because I think in heart failure in general and also genetic cardiomyopathies, in the end, the switch from glucose to fatty acid is happening there as well. So I try to understand why this is so different.
Because there are several mechanisms involved in that process. So we didn't explore the genetic underlying causes, but we know that, for example, there are several proteins that are dysregulated in this process. About the genetic underlying causes, I was not into that, so I cannot tell you more, but for the other causes, there are several proteins that are dysregulated.
But basically, you answered already a little bit what I hoped for, because you suggest there are multiple mechanisms. So what I'd like to explore now a few minutes is what is the chicken and the egg? So your study suggests these are the causal effects, and I would like to debate with you whether it's true or not, and whether that not are like responsiveness of things happening in the whole organ. So one detailed question that I would like to address to you is the central role you put CD36 in the beginning of your thesis and throughout all the figures, you see that, but also in your animation now and also on the discussion so far, I didn't hear it coming back. So how do you feel that CD36 plays a role here in this mechanism?
Yeah, I understand your question. So what we see is that so in this mechanism, CD36 is the primary cause that initiate all these mechanisms. So we see that indeed with excessive fatty acid, it caused this disassemble of v-ATPase, and what you saw in the animation was the fatty acid transporter that was indeed CD36. So this plays like a big role in this picture, because it starts all this cycle of lipid accumulation into the heart, at least what we always observe in our models and in our research.
And did you look at the localization of CD36 throughout your experiments? So you used animals, different rodent cell lines, human cell lines. Did you look at where they were and what happened?
Yes, we also look at that, and it's like several years that we established this model. So it's mostly like distributed between intracellular compartment and plasma membrane. So it's like around 50-50, and then under pathological conditions, such as, in this case, fatty acid oversupply, it will like relocate more and more to the plasma membrane. So we also look at that.
Mm-hmm. Did you ever hear about the soluble CD36?
Sorry? Soluble? Did
you ever hear the soluble, yeah, so the not membrane bound?
Now I don't recall that. It doesn't come in my mind. What I know, what I observe is like between membranes, but yeah.
Yeah, well, it's not so much about the soluble CD36. It's more about where the CD36 can be localized. It is well known that it is associated with different metabolic syndromes. It circulates around, comes from different cell types. And where I want to go is, because I was triggered by one of your figures on page 48, where you suggest as well that Rab proteins are involved. That's figure number 2. You don't have to go into detail there. That's also for me, just that I saw it early on. So Rab proteins is involved in EV signaling, the extracellular vesicles, how they are produced, how these vesicles are moved around in your cell. And also, if you look at your table from chapter 4, about page 110, that's a huge list of proteins. Also, Dr. Hedder, what he was referring to, it's so many targets, so many things that are happening there. But if I look at the names here, many of them are involved in EV biogenesis. And then I refer to these small extracellular vesicles also released by cells and how they propagate signals and how the communication works. Did you think about that? So that also your mechanism partly is involved in cell-cell communication between different cell types, but also inside the cell itself, how this translocation works, and that many of your results you saw here can be like a misplaced balance kind of on that level.
Yeah, it's an interesting question. It's also what interested me, because this process, so I think you refer to palmitoylation indeed. Like this process is involved in many like mechanisms. So other than glucose or fatty acid uptake, indeed can be involved in extracellular vesicle production or communication between cell, and this metabolic disbalance can also influence that. So, and I think that palmitoylation, it's really a signal that it's like a sensor that can sense this metabolic alteration, and maybe also in the AV production is involved for sure.
No, I completely agree. I hope I still have time for a short question, because I want to like a new hypothesis to you, and you challenge me for that. So you provided glucose in early experiments, and now you have amino acids to rescue your phenotypes and your disease mechanisms. What if this supplementation is not just a scaffold or a decoy that you add to the cell, and thereby you normalize the balance again? ATP production, the levels are getting normalized. And not so much about the mechanism of the different molecules, but basically you bring in an oversupply that takes away this over amount of fermentation of other proteins.
Can you be very brief in your answer?
Yeah. I don't know if I followed completely, but what I understood is that we give the cell amino acid, but then it's not that we oversupply, but then we start to rescue again the uptake of glucose that is outside the cell, that is kept out of the cell, and so it is flowing in the glucose that the cell, let's say, need, and it's not like an over amount of glucose inside the cell on what is there anymore. I don't know if I answered your question, but this is what I understood.
Close enough, and I should be in Maastricht to debate further through the reception. But I'm really happy with your answers and for the open discussion. Thanks a lot, and giving the word back to the chairman.
Thank you, Professor Sluiter. The opposition will be continued by Dr. Steinbusch. She is an expert in inherited metabolic diseases at the Department of Clinical Genetics at the Maastricht University Medical Center. Please.
Good afternoon. Dear opponent, thank you for inviting me to your dissertation committee and to this afternoon. And I remember when we met, when you just started your thesis and we discussed where to find all the stuff in the, in the fridge, in the lab. And then this turned out into this huge project. So as mentioned, I work at the genetics department now, so I want to go to the genetics. And I was wondering what you know from the genes that code for the subunits of v-ATPase that you've studied, and what you know of patients that have variants in these genes. Did this help you to form your hypothesis, or can this help you to form future research projects that follow up on this thesis?
Esteemed opponent, thank you for your words, and to remind about when we met the first time. So yeah, indeed, this is very interesting. It's also why this project started at the beginning, because also we wanted to look into that. So if there were mutation of genes of this large subunit also in patient with diabetic patient, for example, and this would explain maybe the not functioning of this protein, or if this protein is more subjected to like oversupply of fatty acid or not. I don't know about any genetic mutation about that in the heart, but yeah, it's really interesting to look at that.
Well, yeah, maybe vice versa. You've collected a lot of knowledge. You've collected a lot of data on the functioning of v-ATPase and of the functioning of the different subunits. Do you have an idea how this can help the known patients in maybe treatments or follow-up therapy?
Yeah, so you mean if, like, look at single subunit of v-ATPase?
Yeah, or the amino acids that you propose, this cocktail, can this be useful for patients with a defect in the A2 subunit or the B2 subunit, the V1B2 subunit?
Yeah, it's a really good suggestion for future work. I think also Joost would be happy with that. But yeah, it would be interesting to see indeed for like maybe mutate each subunit and see what amino acid cause, and which is really the very important subunit that contributes to the effect of the amino acid, for example.
Yeah, and what I find puzzling is that when you look in the literature or in the patient registries, when there's a defect in the V1 subunit, there are not so many symptoms, there are not so many problems for the patients, but when you look at the V0 A2 subunit, there are a lot of things going on. Did you notice that as well in your studies, that subunits were differently affected upon a high-fat diet or high-fat medium in your cells, or was it the same? Can you relate this to what we see in patients?
So in our experiment, no, we didn't do that like a single subunit how fatty acid affect this. So. But for example, our colleagues that did with me the chapter 5, that now is doing experiment using some mice that are mutated for the V1 or the V0 subunit, and at least what he sees is that there are problems anyway, even if you mutate V1 or V0. It's a specific V1G, I think, and also V0D. So he sees that there are problems. But
they are similar.
Yeah.
Yeah. Okay.
Yes. Yes.
Yeah,
Yes.
like
Yes.
in your studies.
It is.
Well, thank you very much for your answers.
Thank you, Dr. Steinbusch. The opposition will be continued by Professor Goossens. His chair is Cardiometabolic Physiology of Obesity at this university.
Thank you. So, dear candidate, I've read your thesis with a lot of interest, and what I in particular appreciated is that you try to unravel the mechanisms underlying v-ATPase dysfunction and its role in cardiac lipid-induced insulin resistance and contractile function. So I would like to congratulate you with the work performed, and of course also the supervisors. Congratulations with that achievement. Of course, I also have some questions to follow up on my colleagues. And my first question actually relates to proposition number 5, and that also relates a little bit to maybe dietary strategy. So I would like to ask one of your paronyms maybe to read aloud proposition number 5, please.
Yep. The heart is flexible and highly energy-demanding metabolic organ. A balanced intake of glucose and long-chain fatty acid over time is essential for its proper functioning.
Thank you. So especially that second part of the proposition, a balanced intake of glucose and long-chain fatty acids over time is essential for its proper functioning. So just to clarify, do you mean that cardiac function will remain normal as long as that balance between glucose and fatty acids is maintained?
Highly esteemed opponent, thanks for your question and for the kind word for the thesis. Mm-hmm. Yes, so the answer is simply yes, that if there is this balance over time, the heart can produce the right amount of energy to function properly, to beat properly, to produce ATP in the right way to function.
Okay. Now then I would like to challenge you a little bit. If we think about a situation of prolonged positive energy balance leading to obesity, right? Let's say that there is a situation of excess caloric intake. And let's assume that the balance between glucose, carbohydrates and fatty acids is still maintained in the diet with excess calories. Is it then still okay? You don't have any problems with cardiac function then, or?
This is a really interesting question, and this is a debate also. So of course, together with the balance, there needs to be also the right amount of nutrients. Mm-hmm. Because if you anyway overload the heart, but any other organs in our body, mm-hmm. there will be problem for sure. So I think it's together with the balance, maybe I should have also add the right amount, because also that is important.
Okay, because well, the question is, do you know what happens when people develop obesity? It's indeed ectopic fat storage. That's what you mentioned in your thesis, right? Lipids will be stored in other tissues than adipose tissue. But there is also something going on with respect to inflammation, right? Low-grade systemic inflammation. And can that also impact cardiac function?
Yeah, yes, for sure. And we also the follow-up work of our colleagues looked into that. So it looked also in inflammation in the heart. And what we saw, it's like that increasing fatty acid cause more inflammation because there is more translocation of CD36 together with it binds to a receptor which is the TLR4, I think it's called, and this can also increase inflammation and fibrosis in the heart.
Okay, and you just mentioned it's maybe I should have added the amount of calories, but you still said that balance is important. So what if I say that caloric intake is more important than the balance? So if we think about prevention, treatment, let's say patients with obesity that don't have developed cardiac dysfunction yet, but we want to prevent complications, right? So is caloric restriction then more important than keeping that balance between carbohydrates and lipids? Is that more important?
Honestly, I don't know the answer because it's a really big question. It's a really big issue. But in my opinion, it's both. both the balance and the caloric intake. And maybe the caloric intake is more, in my opinion, is more important for other organs than the heart.
Okay, we can discuss that a little bit later, I think, during the reception. I'm not sure if I agree with you. But to follow up a little bit on dietary strategies, and I was also triggered by chapter 5, and that was already discussed there, where you suggested that amino acid mixture that might be beneficial with respect to contractile function and cardiac lipid-induced insulin resistance. What about if we talk about lipids? It's not only about, we're talking about calories, and it's also about the quality of the lipids. So what about unsaturated fatty acids? A strategy where you would supply or advise a diet which is rich, high in polyunsaturated fatty acids. Would that be, do you know if that has been examined, what the effects on, maybe whole body physiology, but especially also on cardiac function are of PUFAs?
Yeah, thanks for your question. I don't know exactly, but what I know, for example, that PUFA or MUFA, they lower the receptor on the liver for the LDL particles, I think. So there is decreased in the LDL particles. So I think it can be beneficial also for cardiac functioning, because there is less cholesterol like in the body that build up. So
yeah, I think there are multiple beneficial effects. Also in adipose tissue, it can lower inflammation there. We just talked about the importance of that. And I think also in the muscle, we did some studies in our group showing that it's not the diacylglycerol or the ceramides in muscle causing insulin resistance, but rather the saturated fatty acids, which you might be able to reduce with that. So that might be an interesting strategy. Do I have time for a small question? Then very briefly, I would like to go to chapter 4, a very interesting chapter in which you also showed that pharmacological inhibition of general protein palmitoylation prevents insulin resistance and contractile dysfunction in these cardiomyocytes. And you also actually try to identify specific palmitoylated targets that might be involved in the onset of insulin resistance. And you actually came up with the insulin-regulated aminopeptidase IRAP. And you also described it in your thesis that in fat cells, as well as in muscle cells, IRAP has been shown to improve insulin-stimulated glucose uptake. Whereas in your chapter 4, you actually show that there are beneficial effects of IRAP knockdown on glucose uptake in cardiomyocytes. So that's, as far as I see, the opposite, right? So how do you reconcile your findings with effects in other tissues? And maybe you have partly addressed that in previous questions, but how to reconcile that?
Yeah, that's also an interesting part of the work that I've done. I was really interested in that because, so what I think is that what is also showed that different tissue express different DHSC, so different palmitoyl acyltransferases. And different palmitoyl acyltransferases can palmitoylate the protein in a different way, and depends also on the function of the protein in that specific tissue. Yeah. So for example, in the heart, maybe palmitoylation is like more palmitoylation of IRAP indeed decrease the uptake of glucose, and so it's a regulator in this sense in the heart. Maybe in other tissue is.
Yeah, so we hear that a lot in this discussion, tissue-specific effects. So thinking about strategies, I think you studied the heart pretty much in isolation. I think you need to move forward. We should also look at whole body physiology. And
yes.
So I'm very happy with your answers, and I would like to give the word back to the program.
Thank you, Professor Goossens. The opposition will be continued by Dr. van der Vorst, and he will finalize the opposition. He's from the pathology department at the Maastricht University Medical Center. Dr. van der Vorst.
Thank you very much. Congratulations on this very nice thesis. Also congratulations to your supervisory team. So I enjoyed reading it. It's not my topic, so I learned a lot. And I also had some questions, of course. So I want to start with chapter 4. So in chapter 4, you use red cardiomyocytes as well as HI1 cells. And then using this 2BP treatment, you see that you can preserve the insulin signaling in the red cardiomyocytes. Well, this is not possible in the HI1 cells. Can you speculate where does this difference come from of these effects of 2BP?
Esteemed opponent, thanks for your question. And so what I can speculate is the nature also of this cell. So HL1 cells resemble more neonatal cardiomyocytes, so they rely more on glucose for their survival. So also when you're culturing, you have to give them glucose in order to survive. So it's really a cell line. So I think that palmitoylation has, like, let's say, less impact on the function of this cell than on cardiomyocyte, that they use more fatty acid for their survival anyway, and also palmitoylation there can be more important as on the functional side of these cells. Also, to come back for the expression of the enzyme, can be for sure that maybe in the HL1 there is, we didn't look at that, but maybe in HL1 there are less enzyme, less DHSCs expressed, and palmitoylation has less impact on these cells.
So, following up on this, I was very surprised that, so I understood your reasoning why you then use the HL1 cells to do the DHAC knockdown experiments, but I was actually surprised that you then do see effects with this DHCC or DHHC5 knockdown. So if you argue palmitoylation doesn't really have so much effect in the HL1 cells, why does the knockdown of DHHC5 then have an effect?
In HL1?
Yes. Because you're then also manipulating this palmitoylation.
Yeah, so you refer at the result on the glucose uptake in HL1, no? Yeah, because indeed they also rely also on glucose. So it's possible that these results are because of that, but we see the same in rat cardiomyocyte. So HL1 is, we used more like a model to knock out, and then we report like functional finding in adult rat cardiomyocyte.
Okay. And then following up on this DHC5, so I think in figure 7 in the chapter you then conclude that this DHHC5 is responsible for the hyperpalmitoylation. It's on page 94.
Yeah.
But as far as I understood, you're actually then measuring this at baseline, so you don't add palmitate into these experiments. So how can you then speak about hyperpalmitoylation if you don't add palmitate? Or you're not looking at baseline? And do you expect differences between these two conditions?
Yeah, thanks for your question. I understand your question indeed. So, yeah, this experiment were done indeed in HL1, where we knock out the DHSC5, and they don't need palmitate to survive. It's also a follow-up experiment that we wanted to do that because indeed you can do more causal effect on, you can conclude more from that if you do in high palmitate also. So yeah, it's really a good suggestion for a follow-up experiment on that.
Yeah. So do you time another intriguing question? So I want to switch to chapter 5 where you use these KLR treatments, so to supplement with these amino acids. I'm actually intrigued that you already say that you want to start treating patients, because if you treat mice with this, you actually see a massive upregulation of plasma lipids in these mice. And in the conclusion, you say that this was expected. But did you really expect this amount of increase in the lipids?
Thanks for your question. And so, of course, it's, I think the amount of amino acid that we use also needs to be tweaked in that sense. So we kind of expected, because our reasoning is that giving amino acid would take out fat from the heart and from also, I think, skeletal muscle, because the mechanism could be the same. So in a way is expected, but if you want to apply from like a clinical point of view, I think you have to tweak more the amount of amino acids. So maybe we used like an extreme model to look at the mechanism. So that's why we also wrote that was expected, because other than the heart, maybe also these things happen in skeletal muscle. So
I was actually surprised that the effect is much bigger than the hyperdiet effect. Do you think, is it purely lipids that are coming from the tissues, or do you also get effects of these amino acids in the liver or in the lipid uptake or in different organs? Did you ever look at this?
No, we didn't look at that. But yeah, for sure it's something that you should look if indeed you want to like apply this as a treatment. It's yeah.
And if you're then thinking about this treatment, what kind of downsides could you then imagine, or are there certain patient groups that you rather would like to exclude from your clinical trials?
This is a good question. Yeah, I honestly don't really know the answer of that, because I don't focus a lot on that side, to be honest. So for that, if we want to continue with the clinical trial, for sure I have to look more at the group of patients.
Mr. Schianchi, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and that of your defense, and I request therefore you and your company to await the results of our deliberations and our return in this room.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side. 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Pretty quick.
Mr. Schianchi, the degree committee here present has assessed the quality of your thesis and the quality of your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Glad is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Okay. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Francesco Schianchi, the degree of a doctor and grant you all rights attached by custom and law. As evidence of this, I now present you the degree certificate, signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Wow.
Dr. Francesco Schianchi, congratulazioni e complimenti.
Grazie.
Congratulations with your degree. But of course, there's more to say than just congratulations. So we will now have the laudatio, which will be given by your main supervisor, Joost Luiken.
Dear Dr. Schianchi, dear Francesco, I'm proud to congratulate you with obtaining the predicate doctor. First of all, I love Italy. It's one of the most beautiful countries in the world. It has beautiful cities, beautiful coastlines, beautiful girls. However, it's not correct to say that in these times anymore. But beautiful mountains, especially the Dolomites, as you know, my favorite hiking destination. Let's not talk about the less favorable subjects as Italian football, the national team.
No, please. No, please.
Or politics. This is a good time to reflect on your PhD period. You started in September 2018 and left at November 2023. So that your total Maastricht period was pretty long, more than 5 years. This extension was partly possible due to Corona money. During this time, we went from lows to highs and often back and forth. In scientific terms, your project was on the role of palmitoylation of v-ATPase in a diabetic heart. Not something easily explainable to your grandmother. Moreover, our lab had no experience how to study protein palmitoylation. Hence, you had to set up the methodology from scratch. Notwithstanding, your start was really promising. You rapidly set up a method to study palmitoylation, a method called acyl biotin exchange, ABE. This was also made possible by your initiative to ask advice from another lab in Italy. With this ABE method, we started studying the palmitoylation of v-ATPase. Then came an absolute shock.
Yes.
Yes.
A bit.
We detected nothing. Nothing. No palmitoylation. What the Okay. Hence, our project hypothesis was immediately killed. Shut down. In Italian terms, this was really vaffanculo. Then we had to regroup. Also, the corona time, when the lab was closed, gave us the chance to reflect on the project and change the plan. Maybe we should investigate palmitoylation of other proteins. But which? For this, we turned to the Maastricht facility in Maastricht, guided by Freek Bouwman. I don't know if he's here. And with quite a bit of effort, we succeeded in unraveling the so-called palmitoylome of the heart. This is a complete list of all palmitoylated proteins. We still have to publish this list, but it will definitely be of great help in the scientific community. Then onwards. From this list, we selected a few proteins of our interest and successfully studied the role of their palmitoylation in the diabetic heart. All these experiments, more than 4 years of work, are now bundled into a super manuscript, as I may humbly say, that we will submit hopefully in the coming weeks.
Couple, yeah? Okay.
All this time, I enjoyed your presence in the group. Besides your scientific skills, you are a great guy to have around. Perhaps a bit chaotic, but always, almost always, in a good spirit, with a great sense of humor and great social skills. You are always ready to give a helping hand to a team member in need. I really enjoyed our little chats in between the experiments. Now to the conferences. We went to several conferences together, nationally and internationally. This was always big fun, and I remember that at the social event of one of these conferences, you were controlling the dance floor with some cool moves. The last conference together was in Seoul, South Korea. However, this conference visit started on the wrong foot because our flight was delayed and we were forced to spend the day in Abu Dhabi. But despite that it was really hot, we had a fun day also together with Miranda and Job. Next, we shared a room at the conference venue to save costs.
Nice.
But this was not a good idea because you made a lot of noise at night. Nonetheless, you delivered a great presentation at the conference, and afterwards we traveled around in Korea for another few days, including a hiking trip in the Seoraksan National Park, which was great fun. I forgot the name of the French guy, an airline pilot who joined us that day, but maybe you remember.
I was not joining that hiking.
Yeah. It was the first day.
But I think you told me the story. Like,
I have to look at the pictures. Okay. However, at return from the conference and towards the end of the PhD period, things took another turn. You had to break up with your girlfriend, which caused you much pain, and you took some time off to return to your family in Italy. Then, as good as bad as it went, you finished the last experiments, also together with our new PhD student, Jeroen. There he is, who will continue the palmitoylation work. Now it's time to wrap it up. You quickly gained a new job as postdoc at the Blood Bank Sanquin in Amsterdam. Meanwhile, you're working there already for almost a year and building up a new life. Still, you left a hole in our team, and I really miss you having you around. Francesco, now it's Dr. Schianchi, I wish you all the best, and hopefully we stay in contact.
Thank you, Joost.
Okay.
Dear Dr. Schianchi, it is my honor and pleasure to congratulate you on behalf of the University of Maastricht with the degree you just achieved, you just acquired. I also would like to congratulate your supervisory team and, of course, your family that came from Italy, Papa and Mama, your friends that have joined us today, and also those that have collaborated with you on the different topics that you describe in your thesis and in some of your publications and publications to come, I understand. You've done a great job in setting up the research, a research line, in a new research line in the lab of Professor Glats and Dr. Luiken. And you performed an extensive amount of work that has been described in your thesis. And also about the defense of your thesis, we agreed within the committee that you did this, you defended your work in a proper scientific attitude, only being sure about things that you are sure about. I understand that you have a postdoc position now in Amsterdam, extending your stay in the Netherlands for another 2 or 3 or 4 years, whatever. So that means that you're now in the Netherlands for 7, 8, or 8 or 9 years.
9.
9 years even.
9, yeah.
And that you must be missing what I presume is the Parma region. Is that correct?
No, this is not Parma, but it was from one of my trips last year.
No,
I really enjoyed this picture.
Yeah,
so,
from your CV, from your curriculum vitae, I understand that you're very proud of being a Parmigiano, or
Parmigiano. Like,
Parmigiano, yeah, like the cheese.
Like the cheese, it's easy to remember.
Yeah, so I would like to wish you a lot of success in your new job and as you call it yourself, in the new molecular world. So congratulations again and lots of success. Ladies and gentlemen, before I close this ceremony, I have a few announcements to make. What we will do first is make a picture here in the room with the candidate, with the doctor. Sorry, with the new doctor. Maybe Papa and Mama can join us here so that we make a picture also together with the opponent online. I would like to ask the audience to leave the room and to wait for us at the elevators. Actually, you can go to the rafter already, because we will join you then after we have made a picture, another picture at the stairs in the hall of this building. Having said that, I can close this ceremony.
Chiudi. Poi mano sotto.